Hello, everybody. You are welcome to this evening's the third Nuclear Masterclass webinar. It's great to see so many of you joining live. I think we're approaching 150 participants, which is great to see on this Wednesday evening. Tonight, we're going to be exploring the very exciting and cutting-edge topic in nuclear physics, which is its medical applications. So if you haven't joined before, you're very welcome this evening. I'm Dr. Elizabeth Cunningham. I'm a former nuclear physicist and science communicator. I am a lover of Lego and its use in communicating science. I am extremely excited to be with you here tonight to chair what I know is going to be an extremely interesting webinar. And tonight we'll be finding out about a new type of cancer treatment called targeted alpha therapy and discovering how we can ensure that it is safe and correctly prepared for patients. And our wonderful speaker this evening is Dr. Katie Staunton-Mann, who is a research and development physicist. She works in partnership with a nuclear medicine company called LabLogic in Sheffield, and also in partnership with the University of York. So I'm very interested to hear her speak tonight. So I'm going to hand over to Katie, who can tell us about making targeted alpha therapy possible. Thank you, Katie.
Thank you, Elizabeth. Thanks for that introduction. So today I'm going to be talking to you about targeted alpha therapy and how quality control is making that all possible. But first, a bit of who am I, what am I doing here, how am I got any sort of expertise in this? Well, originally I started off studying physics at University of Leeds, and then I decided to somewhat leave physics behind and go and do a PhD looking at developing bone and looking at how to recreate that at the nanoscale. And then afterwards I decided, no, actually I do want to go back to physics. I'm really good at that. I enjoy that. And I found a job at LabLogic. LabLogic's a company that specialises in detecting radiation and specifically detecting radiation in the nuclear medicine market and industry. And so this position was actually joint with the University of York, so I'm a partner in between both. And really, the whole project is about developing quality control instruments for alpha radiopharmaceuticals or targeted alpha therapy. So I'm going to go a bit more into that now. But before I get to targeted alpha therapy, I'm going to go back more to the basics. So what is an element and what is an isotope? So this is the periodic table. I'm sure you might have seen it all before, but we're just going to talk through it again. So here you've got all your different elements, and then each element is decided by the amount of protons that's in its nucleus. So that is what defines an element, is the number of protons in its nucleus. But what defines an isotope is the combination of the number of protons and neutrons. So here's a fluorine. It's got 9 protons, but it can have a variety of different neutrons in there, which means that this number at the bottom isn't a whole number, because you don't always get 19 total protons and neutrons. You can have 18, you can have 17. So this number then changes the behaviour of that isotope. We're going to look at this, is called a nuclide chart. And this displays all the known isotopes. So as you go up in each row, you're changing element. So at the bottom, you've got, in the bottom left, you've got hydrogen, and the top right, you've got some very heavy elements. As you go along each row, you're changing the number of neutrons that are in that isotope. And so further towards the right, you have increasing numbers of neutrons. You can see here there are some different colours on this chart, and they refer to the radioactivity and decay routes that these isotopes will undergo and to get stable. So in the black, these are all the stable isotopes, which means they're not going to emit radiation to become stable. But on either side, different radiation decays are chosen to kind of get towards getting to that stable isotope at the centre. So these isotopes can undergo, maybe if they're quite close to one of those black stable isotopes, just one decay to become stable, or they can undergo a few different radiation decays before they become stable. So when I refer to, say, a parent, that means the original isotope that we're talking about. If I talk about a daughter, that means it's produced from a radioactive decay, but it's not yet stable. So it will also undergo decay. So there are a few different radiation types that you might have heard of. So there's alpha, beta, and gamma ray radiation. So alpha radiation is actually just a helium nucleus without the electrons. So it's got 2 protons and 2 neutrons, and this makes it pretty heavy and also highly charged. So it doesn't get very far. When you think of a smoke detector, it's the whole basis of it is that the alpha particles cannot pass through smoke. Smoke is enough to stop the alpha particle, to stop the current going through. So here you can see that an alpha particle is stopped by paper. Whereas a beta radiation, this is a positron or electron that's been emitted from a nucleus, and it's stopped by some aluminium. Whereas gamma ray radiation, this is electromagnetic waves or photons, and this can pass through paper, aluminium, lead. It can penetrate quite far. So their penetration and the way they interact with matter, these different radiation types, is how we kind of choose how to use them in nuclear medicine. So gamma rays can pass through matter quite easily, and so therefore if you're trying to see inside a patient, you might want to choose a radioisotope that emits gamma rays because it's going to reach your detector, whereas an alpha or a beta probably won't leave the body, and in fact they definitely don't. So alpha particles can actually only travel about 1 to 3 human cells. So they are interacting so strongly that they will stop very quickly, and along the way they cause a lot of damage. Beta particles can travel quite a bit further, they're more up to about 1,000 cells, so they can cause some damage, and they do cause damage, but it's far less localized compared to alpha radiation. So as I said, like, alpha particles can't get very far, only a few cells, and in that process they're damaging the tissues that they're around by causing ionizing radiation. So they can cause this damage to tissues in 2 different ways. So you've got direct damage, which is through double-stranded DNA breaks. So they will basically just rip apart DNA, and when a cell doesn't have DNA, it dies. So you're killing a cell that way. In indirect damage, where basically when this radiation is passing through matter, it's knocking off electrons of different molecules, and this creates free radicals, and this is basically charged molecules, and these then go and attack different parts of a cell. So for example, that can cause damage to DNA, and this will also cause a cell to die. So at the moment, alpha radiation seems quite negative, actually. It's just causing a lot of damage. So why is it being used in therapy? So the idea is, with an alpha radiopharmaceutical, you have this alpha-emitting radionuclide that's going to be emitting alpha particles. So when it is emitted, it is going to be causing damage around. So you want to make sure that that is targeted to the bits that you want to kill. So that's where targeted alpha therapy comes in. And here what we have on the left is a kind of schematic, an idea of how these targeted alpha therapies look like. You have the alpha-emitting radionuclide that is bound by a chelator. So this kind of holds it in a bit of a prison almost. And then the chelator is connected to a vector, and this vector is chosen to specifically target certain biological targets that are found on cancer cells. So these targets are overexpressed on cancer cells, such as PSMA, which is found on the prostate. So you produce a vector that fits in that like a lock and a key. And so when you put this injection inside of a human, it's going to find all the PSMA, and it's going to attach to that. So when that alpha emitting radionuclide decays, the alpha particle is very close to the cancer cell and will pass through the DNA and kill it, hopefully. It only takes a handful of alpha particles to kill a cell, so you don't need that many to really get an extreme therapeutic response. So on the right-hand side, we can see this is one of the very first times targeted alpha therapy was used in the clinic. And on the left, we can see a imaging and a PET image of a cancer patient which has a lot of different tumours, unfortunately. You can see the body's quite riddled with it. It's very late stage disease. This person was then given an actinium therapy with PSMA. And afterwards, you can see that a lot of those tumours, the dark spots, have gone away. They've reduced. And this was really the power of targeted alpha therapy, that it was able to seek out those tumours that are extremely widespread across the body and can kill them quite effectively. So that's all well and good. It has a great potential, but how is it actually going to be applied in the clinic? Because it can be quite dangerous, as we've said. It can kill all sorts of tissues. If it's not targeting properly, it's going to kill a lot. So before we get to the patient, the radiopharmaceutical goes through quite a few different processes. So firstly, the isotope has to be produced. This can be in a variety of ways, through accelerators, cyclotrons, through generators, but the isotope in some way has to be produced. This isotope is then transferred to a synthesis hot cell, and that's where you have a synthesis unit, and this is where you're effectively doing the chemistry, binding that isotope onto your targeting molecule, or incorporating it as part of a sugar molecule, which is what FDG used in PET is done. And then that is then transferred, your final radiopharmaceutical product is transferred to a hot cell where it's then dispensed. So this is where you go, what dose does this patient need? What activity do they need? How much they need based on different requirements? So that's where it's dispensed out. But before it's given to the patient, it has to pass through quality control. So quality control is the last stage before it's given to a patient, just to make sure that everything is fine. So there's quite a few quality control tests. These are all conducted by trained radiochemists, so they're people who are trained to work with radioactivity, but they're also chemists as well. So it's quite a niche role in between a few different disciplines. And these quality control tests are determined in regulations. Before anything can be given to a patient, there's a monograph is written, and they determine the different quality control tests are required to make sure something is safe before given to a patient. So some of these tests include just appearance. So you actually have to look at it with your own eyes and say, is it clear? What colour is it? And then you have to check the pH, because obviously the body exists at a certain pH, and there's only certain levels of pH that can be given to you and it still be safe. So if it's too acidic or too basic, it can't be given to a patient. So that's tested. And then also these radiopharmaceuticals are developed using lots of complicated chemistry, and so that might include solvents that aren't very good for humans. So the amount of residual solvent has to be detected, and it can't pass a certain threshold. If not, it can't be given to patients. We also want to make sure everything's sterile before it's given to a human. So you have to undergo these bubble point tests, which are effectively, you filter and sterile. You make your radiopharmaceutical sterile by pushing it through a filter. You have to check that filter's then it's got its right integrity, so you use a bubble point test for that. You also have to check if there's any endotoxins, which are sort of like bacteria or viruses that may be inside your radiopharmaceutical. That takes quite a while to come back, so that's actually a test that's finished and you get the result of, say, 2 weeks after it might have been given to a patient. But that's purely due to the nature that you can't get the results fast enough. Because in this whole process, after you have produced your radiopharmaceutical, it's decaying away. It's undergoing radioactive decay, and that is exponentially decreasing the amount that you have. So you want to get through this QC process as quick as possible. And that leads me on to checking the amount of radioactivity that you've got, because your dose in nuclear medicine is based on activity. It's not based on concentration necessarily. It's the radioactivity concentration. You also want to check the radiochemical purity, the radiochemical identity, and radionuclidic identity. I've jumped over those a bit quickly because I'm going to go in a bit more detail because that's mainly where I have focused with my work. So with radionuclidic identity, the question that this is really answering is which isotopes are present. You might have the isotope that you actually want, that you want in your dose to your patient, but there could be other isotopes in there due to impurities in the manufacturing process, and some of them can be quite dangerous, and so we need to make sure that if they are present, they're present that's considered acceptable at very low quantities. So how do we determine radionuclidic identity? It's through gamma spectrometry normally. So this is where we place our sample in the gamma spectrometer, and this will give us a gamma spectrum. And these different lines that you see on the gamma spectrum are associated with different gamma emissions, and those are associated with different isotopes. So each isotope somewhat has a fingerprint that you can say, oh, it's got that emission, it's got that emission, it's got that emission. Okay, we know that that's that isotope. We can also look at the half-life of these isotopes, so how quickly the activity decays away can be used as a method to determine the different isotopes that are present. But if you've got multiple, that might complicate it, so gamma spectra are used. Something to consider with the gamma spectrum, here you can see that the peaks are quite nicely resolved from each other at different energies. But that is because that gamma spectra is taken on quite a high resolution gamma spectrometer. And when I say resolution, that means the full width half max of the peak. So this is where you have the height of the peak, we take it in half, and we determine the width of that. And that is given our full width half max. So that spectrum is taken on a CZT, cadmium zinc telluride, and that's got a resolution of 2.5%. But if you were to use a sodium iodide gamma spectrometer, you'd have a poorer resolution. That's 9%. And so you might not necessarily be able to resolve those different peaks because they broaden and they overlap. So we also have radiolabeling yield, and this is part of the question for radiochemical purity, really. It's just two different tests, but they have different names. So radiolabeling yield is answering the question, how much is attached to a label? Because all of those free isotopes that you have are going to be dangerous to a human because they're not going to be targeted anymore. They're just going to cause alpha radiation that's damaging your healthy tissues. And we really don't want that. So we want to make sure as much as possible it's labelled. And the minimum requirements for this is set out in the monograph I was speaking about. So normally it's 95%, potentially even higher, potentially 98%. It really depends on each isotope and the damage that it could cause if it wasn't attached to the label. So there are different reasons as to why it might not be attached to a label. One of them is that, say, when this radioisotope undergoes alpha decay, the alpha can go in one direction, and that causes, with the conservation of momentum, for the daughter isotope to go in the opposite. And this process is far more energetic than the chemical bonds keeping the isotope in place by the chelator. And so it gets ripped apart, and this process is called recoil. So we need to know how much of those free isotopes are potentially around in the radiopharmaceutical, but also some of them may just have never attached in the first place. And so that's something we need to determine. And the way we do this is by determining the radiolabeling yield. So we have what's called a thin layer chromatography strip. It might be similar to if you've done in your chemistry classes with paper and separated out a black pen. So what we do is we put a piece of sample onto the thin layer chromatography strip and we separate it out with solvents, and the labeled and the free components travel different distances along the strip, and this is due to their interactions with the TLC strip. That TLC strip, once those parts are separated out, we place that on a TLC scanner, and that can then pass a radiation detector over the top of the strip, and it determines where the radiation is along the TLC strip. From that, we can see there was more radioactivity in one place or the other. This is effectively a chromatogram, which is the amount of counts per distance. And we use the area underneath that chromatogram to determine the radiolabeling yield. So the radiolabeling yield is the amount of labelled divided by the total activity. So radiochemical purity. This is slightly different to radiolabeling yield. It's because it's answering the question, is the label targeted? So during all these alpha decays that are going on, we're having lots of ionization events. Those ionization events can then cause radiolysis of the labels on other molecules, damage them, change their structural form. That structure is very important to fitting your target, your biological target. So if that changes, it may not be targeted anymore, and effectively, it's not going to be targeting that vector. That means it's free. It could be damaging healthy tissue. So we need to determine how much of it is actually targeted. And to do that, we need to be able to separate out the product and also radiolized product. For that, we need something that's much better at separating out, which can be with a HPLC. So this is where we use a column on a high performance liquid chromatography, and this separates out the different components really, really well. It then passes through a UV detector to say what chemical form it's in, and then it passes through a radiation detector. And from that we get a chromatogram, and we can get our radiochemical purity. So now I've covered all the background, I'm going to talk about actinium-225, which is the biggest alpha emitter out there in targeted alpha therapy. So this is the decay chain of actinium-225, and it's quite complicated. Here we've got at least 5 potential alpha decays. We have a few daughters that are emitting gamma emissions. We have a parent isotope, and then plenty of daughters that are all emitting different types of radiation. So when we're doing our radiolabeling yield or radiochemical purity measurements, what are we going to choose to do our quality control? What will be our radiation detector? So there are plenty of different types of radiation that are being emitted there, so we need to be sure. So let's look back at that decay scheme again. We have actinium-225, which is not emitting any gammas. It's only emitting an alpha. And gammas are much easier to detect than alphas. They pass through anything pretty much, especially in all the different coverings that you might choose to cover your TLC strip with. For example, people put their TLC strips in plastic bags just for radiation protection considerations. And yeah, the gammas are going to pass through that. So if you wanted to look at the alphas, the alphas will be stopped by that plastic bag. So you'll have to take it out of the plastic bag. Some people make their decisions on which radiation they want to detect based on the radiation protection concerns and regulations in each country. So we're going to look at the gammas for now, because the gammas are much easier to detect. So francium is the first gamma emitter down the decay chain with the significant gamma yield. It has a half-life of about 5 minutes compared to actinium's 10 days, so it's a much shorter half-life, and it has this gamma emission at 218 keV. Bismuth is another daughter that has a gamma emission at 440 keV. It has a bit of a longer half-life, about 45 minutes, and has a yield of 26%. So it's got a bit of a higher gamma yield. So that's the probability that when these decay, that gammas are going to be emitted. So when I say francium has 11% yield, 100 franciums will decay and only 11 will emit the 218 keV gamma. So it's not a very high yield of gammas that are being emitted here. So we've established we want to look at those two gammas, but how can we relate those gammas to actinium? Because actinium's not actually emitting those gammas. So this is where we look at secular equilibrium. So secular equilibrium means that in a decay chain, when you have a decay of a parent, it equals the decay of the daughter. For that to be the case in this situation, that means we need to wait for the daughter's activity to grow in enough that they equal the same activity of the parent of actinium. There's quite a nice, easy equation that we use to work this out, which is effectively get the half-life of the daughter and times it by 6, and that's how long it takes. So we look at francium, it's about 5 minutes, and so we say after 30 minutes, that will be at secular equilibrium. Also, for the bismuth, that takes about 4.5 hours once we times that half-life by 6. If we look at the whole decay chain, we're looking at really about 24 hours for the whole decay chain to be in equilibrium. So if you're waiting for the last daughter to be in equilibrium with actinium, you're waiting a full day to do your quality control tests. So while it's not ideal that francium and bismuth, we have to wait to use those for actinium, at least we're not having to wait for the whole thing to be in equilibrium. So I'm going to just show this again with just the daughters that actually have gamma emissions. This is the francium and the bismuth. In the red lines, we've got francium. So we've got francium that's growing in from actinium as that's increasing in activity. But also, if we had any francium on the TLC strip to begin with, that's been decaying away. That's important because you want to make sure only that francium that you're detecting is from actinium decay and not just because there was some free francium there to begin with. So the same with the bismuth. After about 5 hours, we have the bismuth growing in and reaching the same activity level as the actinium. So at that point we can say, yes, that's at secular equilibrium. And equally, during that process, during that time, or any free bismuth that was there is decaying away. So then we know any bismuth decay that's happening after 5 hours is due to a decay of actinium-225. So here we have a different representation of that. You can see this is an example of a TLC strip with a detector over the top. So you have your peptide or your labelled end and your free end of your TLC strip. So at time 0, you can have actinium, you can have francium, you can have bismuth, you can have a whole mix of these. And then after 30 minutes, the francium that was there at the start has decayed away, the actinium has decayed, and francium has also been decaying, so that's been growing into the same activity levels. And so you'll have a little bit of bismuth 213 as well, but not very much at 30 minutes. And then after 4.5 hours, 5 hours, the original bismuth will have decayed away, and then you can have just the bismuth that has grown in from the actinium and francium decay down into bismuth. So they're all in equilibrium at this point. So if you wanted to get a correct radio labeling yield, you have to wait with your TLC strip. Once it's been developed, it doesn't matter if it was in secular equilibrium before you started your TLC strip, because as soon as you separate out those components, the clock starts again. And that's a concept that people sometimes in the industry still struggle with. So I understand that that's a bit hard to get your head around, but it's really interesting to see how we have to use these decay chains and really think about them really carefully to be able to use that to determine your radiochemical purity measurements. So just to summarise the different detection options that we have for looking at actinium is after 30 minutes, you can look at francium, and that's at 218 keV gamma. After 4.5 hours, you can look at bismuth. But equally, once that bismuth's in equilibrium, you can also look at all the total alphas if that is something that people would like to do. So the alphas, there'll be 4 of them at that point in total. And alphas can have a lot higher signal to noise ratio, especially considering the fact that alphas there are 4, and that yield is much higher than 26% of a gamma. So if you have an alpha detector that you can use, sometimes that is actually much more preferred. But if people choose to wait longer, they can wait up to 24 hours afterwards and then just look at all the total gammas or all the total betas. And that's something that people do choose because actinium has nearly a 10-day half-life, and so they say waiting a day, that doesn't mean that much compared to some isotopes with shorter half-lives. So we're actually going to move now on to an isotope which does have a shorter half-life, which is lead-212. And this is something this isotope's really started to kick off, and there's been a really exciting project in the UK specifically at looking at extracting lead-212 from some of our legacy nuclear waste. So this is material that was just sitting around about to be buried, and now we're potentially using it for cancer treatments, which is incredibly exciting. This project was launched at the end of last year, and it's a multi-year project. And I think in the next few years, we're going to see a large growth in the area of using lead-212 for cancer treatments. If you look on BBC, typing in National Nuclear Laboratory or Medicines Discovery Catapult, you will see some news stories about this, which are very exciting. So let's look a bit more about lead-212 and the decay chain. So lead-212 does have a short half-life. It's 10.5 hours, and that means you've got to work a bit quicker with your QC. You care a bit more about getting your quality control done quickly because every hour that you might take, you are losing patient doses there. So you really want to maximise your patient doses to treat as many people as possible. So here you're not waiting 24 hours to get your quality control results because you want to inject as soon as possible. Lead-212 does have 2 alpha emissions in its decay chain. It itself is actually not an alpha emitter, it's a beta emitter. So those issues with recoil and producing free isotopes are less problematic with lead-212 than they are with actinium, which is quite exciting, and I think that makes it a very promising isotope in this field. But it's actually its daughter bismuth-212 is the alpha emitter. So in a way, you can think inside the human body, lead-212 is becoming a generator for bismuth-212 to do its job and producing the alpha particles, the alpha radiation that's going to be damaging, killing those cancer cells. But as I've spoken before, the alphas we don't choose to use that for quality control too much. And also, it doesn't really relate to the parent isotope in this situation, because with all these quality control tests, we care most about the parent isotope because we say lead-212 is the radiopharmaceutical, and so we need to look at specifically the lead-212. In this case, lead-212 actually has some gamma emissions compared to actinium, so that makes it a lot easier to look at those for the quality control of lead-212. So at 238 kV, it has a 44% yield gamma emission. So that means for every 100 lead-212s to decay, we'll have 44 gamma emissions, which is excellent. But you can see some complications in the fact that the daughters also emit some gammas. So the bismuth 212 has some gamma emissions around 700 to 800, and then also one at 1600 kV. And then the thallium 208 has quite a few different gamma emissions. The 277 kV is quite close in energy to the 238 kV lead-212 emission. And so you might need to start thinking about the resolution. Are we going to be able to separate out those two gamma emissions? And then there are some higher energy gammas that are really quite high yield. So you've got 85% yield at 583 kV, and that's going to cause some problems when we do the QC. So we're going to go through that now. Typically, when you are doing your HPLC, for example, you set an energy window and just look between the energy window, we know that we will capture our gamma emission of interest. So here you've got your 238 kV energy window that's in the center of this, and you've captured that entire gamma emission. So this is a simulated spectrum of lead-212, assuming you've got a sodium iodide based gamma detector. So these are typically used because they're quite cheap and have been employed and used in different for isotopes. They're quite versatile and they have quite good detection efficiency. So this is why you would start with this initially if you were trying to plan to look at an isotope that has a gamma emission. But if we use that energy window and then look at the other gammas that might be arising in that from the daughters, so we have this is the bismuth 212 daughter, and we can see that there have been some gamma. There's a peak around between 700 to 800 due to those gamma emissions. They actually scatter down into the energy window of interest due to Compton scattering. So this is the gamma interacting with matter and losing energy, and then eventually depositing that energy into our radiation detector at a lower energy. If you look at the thallium 208, you have obviously those very high yield gamma emissions at between 500 to 700 kV, and these are going to cause a significant amount of scatter into our lead-212 window. So that can cause a lot of issues with trying to isolate a lead-212 signal. And specifically that 277 kV gamma emission is, it's far too close in energy and using a sodium iodide detector because you don't have the resolution to be able to separate out that gamma emission from your energy window. So just to summarise for the lead-212, energy window, we have both lead-212, bismuth 212, thallium 208, which can all be contributing to the energy window that you would choose to select to investigate lead-212 with a sodium iodide detector. And that means that even if you had no lead-212, you can have contributions from bismuth 212 and thallium 208. So you could have a false positive there. You would be reading, saying you're having radiation coming through and saying you've got lead, but you actually don't have any lead. That's actually the daughters. And also, to be able to separate out your lead-212 and your thallium 208, you need to have a high-resolution gamma spectrometer to be able to separate those. So people have been getting around this issue by using fraction collection. So this means after you run your HPLC, your liquid's been flowing, and you collect that liquid at the end of your HPLC into little vials. So every 10 seconds, you'll have drips into one vial, and then the next 10 seconds, it will move over and drip into the next vial. So for every, you separate out your whole HPLC run into lots of little vials. You wait, and then you detect later. So the reason why they're waiting in this case is you need to have the bismuth 212 and all the thallium 208 contributions to decay away. And this can take quite some time. This can take about 5 hours, 6 hours. Some people choose to look at an earlier time point, but to be sure, some people were waiting 18 hours to do this. And then using that gamma counter energy window around the lead-212, the 238 kV. But this isn't ideal, having to wait that long. If we remember, the half-life is about 10 hours. So in this process, you could be losing at least one, potentially, reduce your activity by half or more during this process, depending on how long you choose to wait. And just also on a personal aspect, I've been in labs where people were using this fraction collection method and they spent hours capping vials, so much so that they had calluses on their thumbs and fingers from just pressing down on vials all day. There's a lot of manual labour and a risk for error when you're introducing that human aspect that you just can't afford when you're doing quality control or something that can be quite dangerous to a person if it's not done correctly. So about this time last year, we embarked on a project to try and eliminate this need. And for that, we developed a product that we've now called Specgram LC, which means spectrum liquid chromatography. And this was combining a gamma spectrometer with a HPLC detector. So what we've done here is we've got a gamma spectrometer and we've placed a flow cell inside the gamma spectrometer. So this then detects our gamma rays that are emitted for down to every 1 second, and we can use this to isolate our lead-212 signal. So I'm going to just show you a video of how that might look. So I'm going to press play. Okay, so here on the top, we've got a total gamma chromatogram, which is effectively all the radioactivity that is being passing through the detector. The bottom, we've got a total gamma spectrum, and this is the energy and the counts that we've got. So the first peak that went through had no lead-212 in it. And then this now green peak that's come through our flow cell has had a lead-212 gamma emission really come up from above that background. So you can see here how we've got two different graphs, one with the total activity and time, and the bottom with the counts and the energy. So what we chose to do was use that total gamma chromatogram, and we really wanted to separate out and just get the lead-212, and we used the gamma spectra for that purpose. So you can see here we wanted to isolate the lead-212 signal above the background because the background was from the daughters and not the lead-212. So this is where we used the gamma spectrum. So what we did was we separated out the entire gamma spectra from the whole HPLC run into little time chunks, and each time chunk had its own gamma spectra, and then applied a peak finding algorithm to look at the lead-212 above the background. So you can see the background slightly sloped here. And that was the idea, to isolate the lead-212 and not include any of the daughter gamma emissions. So if we look at the impact of this, we can see if we use the total gamma chromatogram, we go from a percentage of labelled from 83%, which would be an absolute radiochemical purity fail QC result, no release to patients, you could not do that. When you apply this lead-212 finding algorithm, you achieve 99% radiochemical purity, and that's because these peaks at the start were not lead-212 at all. The region 1 was all the free ions that there could be: lead-212, bismuth-212, thallium-208. And then region 2 was lead-212 impurities, so it was important that we detected those, but it was also bismuth-212 connected to that label, that targeting vector. So we wanted to discount that. And so this shows how the power of kind of combining different physical techniques, different nuclear physics techniques, to really applying that to using in nuclear medicine, combining both gamma spectrometry and simple radiation detection on a HPLC. So just to summarise some of the things I've been speaking about today is that really targeted alpha therapy is a novel cancer treatment that has shown great promise to revolutionising treatment across numerous different types of cancers. But to make sure that it's safe for a patient to have, we need effective quality control. And when we consider the quality control, we need to really think about the decay chain of the isotope we're looking at and the radiation that's emitted to make our quality control unique to the situation that we have. And I hope this has inspired you to think about how nuclear medicine might be changing and how you might see a potential career in it. And it's an incredibly exciting place to be, especially in the UK right now, as lots of projects are going on. So if you have any questions, please let me know. I hope I'll be able to answer them.
Well, thank you so much for that, Katie. That was really interesting, and it was really wonderful to see, this new important cancer treatment and how important it is, how important the nuclear physics that the students in the masterclass have been learning about, how important the nuclear physics is for patient safety and quality control. So that was really nice for everyone to see such an important application to what they've been learning about. So thank you very much. There are lots of questions, so I will head straight to them. So there's a couple of people who are interested in the isotopes and why they have such short half-lives and how that affects transportation of and production of the isotopes. So in the instance of lead-212, how is the parent isotope taken from its place of production to a hospital if they have such a short half-life, and why do they have such short half-lives?
That is a fantastic question, because when selecting these isotopes for nuclear medicine purposes, you really have a balance there of choosing a half-life that's long enough to get it to a patient, but also isn't too long, because once that person's taken that therapy, they're actually radioactive. So in cases where some people have a half-life, some isotopes have a half-life of quite a long time, they might have to be isolated within a hospital and in a shielded environment to protect members of the public from them, because they are a radioactive source at that point. But for lead-212 with a 10-hour half-life, it means that you likely want your place of isotope production to be the same place as labelling. So it will be in the same facility and potentially might be connected to a hospital. But also 10 hours, you can get a flight with that dose. And that is what happens normally, is if you have these targeted alpha therapies, they're normally flown to where the patients are to be able to get them there. Because while 10 hours is short compared to actinium, it is quite long compared to others. So certain isotopes you might have heard more about is fluorine-18, which is used in PET imaging, positron emission tomography. And that is used quite routinely in hospitals and only has a half-life, I think, of around an hour. I don't have it off the top of my head, but it means that it has to be produced locally within the UK and can be shipped and driven to hospitals. But for some of these, which are less local, they are flown around.
Okay, thank you very much. That's really interesting to know that they get flown around. I wouldn't necessarily want to be on that flight with a radioactive isotope, but that's interesting to know. Someone else has asked, is there any way to predict the half-life of a specific isotope without testing it?
Oh, I think it all depends on the stability of the isotope. If it's very unstable, it will have quite a short half-life. And but yeah, it's quite difficult. And some isotopes have very incredibly short half-lives, like nanoseconds. They are difficult to measure the half-life of them. So I was actually at a conference today with people discussing that issue. So
it's topical questions. Still needs some work. Very good. A couple of people have asked about the length that the different checks take on average in order for a treatment to be allowed to be used on patients in hospital.
Yeah. So a typical HPLC run, if it's about 20 minutes. So you'll get your radiochemical purity within 20 minutes. For a TLC strip, you are looking at a similar amount of time, considering the fact that the strip has to be developed. It has to pass through the solvent before being scanned. So these tests can be done quite quickly if you're not having to wait for equilibrium. So a typical isotope that's used for imaging can be done relatively fast. So they are done in a hospital.
Okay.
Yeah.
Thank you. So we've had some questions as well about sort of your career path and your job. So someone has asked, what is it like to work at a company working with a uni at the same time? And someone also asked, what routes should you take to work in industries like this, i.e. should you study chemistry or physics, etc.?
Yeah, that's a great question. So my career path has been very interesting because I went through academia first. I did a PhD, and then I decided that I preferred to work in industry. When I found this role that was in between the two, it kind of kept both of the options open. And I found it's been very rewarding because I've had the expertise of a business who understand the pathway to getting a product to market, and that way it can have the most impact on patients. But we couldn't do that without the academic support and the knowledge that they bring to the table. So it's been really interesting also bringing those two types of people together who have very different drivers and motivations, but we are all united in the common cause that we want this project to work because we know how the impact it can have on patients. And to getting into this industry, I think you could honestly go through both routes. All of the radiochemists that I know all trained in chemistry first, and then they learned how to do the chemistry and the synthesis. And then if they chose to be radiochemists, that's normally typically that's done as a master's program, or some people learn that on the job. So if you prefer to do the radiochemistry, go down the chemistry route. But if you want to be a medical physicist in a hospital, for example, looking at the dosimetry, checking the right doses for patients, and looking at imaging, I would go down the physics route. But I think there might be specific courses for looking at medical physics at university. So that would be something to investigate.
Thank you very much. Someone has asked, what differences do you notice between working sort of in R&D for a company versus research at a university?
So research at university was a lot more long form projects. So when I did my PhD, unfortunately got influenced by COVID, so that delayed it a bit. And I ended up taking nearly 5 years for doing one piece of research. Whereas with a company, they have commercial pressures, so everything needs to be turned out a lot faster. If I said to someone I was going to take 5 years doing a project at my company, they'd probably laugh and say, no, you're not, because we need to sell it to continue to survive. So it's a lot more work on this project, work on that project, be very flexible, being able to pivot at the company, because I have to be able to look at lots of different projects at once to kind of push them all forwards. But also, I would say my time at the company has been very well supported through different engineers. So I'm the physicist in the R&D team. My manager is also a physicist. But we have electrical engineers, we have mechanical engineers, we have firmware engineers, and those aspects are covered by them, whereas I'm looking at more the physics and the concepts behind the products we're developing. Whereas when I was, say, doing my PhD, and this isn't always the same, but I was quite isolated during my PhD. I was the only one looking at my topic, so I had to kind of cover everything. And that isn't the same for everyone. Some research groups have a diversity of people across them, and they all support each other. So I wouldn't necessarily say that was my experience was the experience that everyone has. And it can be very dependent on the research group that you go into.
Okay, thank you very much. Someone has asked, it's quite an interesting question, would the radioactivity levels in a therapy formulation be sufficient to sterilise it without the need for filtering?
This is incredibly interesting and also came up at my conference today, so very topical. It depends on the radiation type. So with alphas, they are very damaging, and so you might be able to consider they would be self-sterilising. For other isotopes that are used in imaging, so they are gamma emitters, you unfortunately cannot assume that at all. So you have to make sure that they are, that they are sterile. But even if you might be able to consider that they're sterile, you still have to go through the sterility procedure just to be sure.
Okay, thank you very much. I think we've got time for one more question. Someone is interested in how do the machines or tools actually detect the particles or rays, and why are gamma rays capable of so much more penetration than alpha or beta particles?
Right, yes. So in the radiation detectors, the radiation will interact with the detector itself. Typically with, I'm going to offer an example of sodium iodide. This is a detector for gammas, and the gammas will interact with the sodium iodide and will produce scintillation light. So it will produce visible photons. Those visible light photons will then be detected by a separate detector, and that signal is then amplified on. But the basis of all of the radiation detectors are that the radiation interacts with them and produces some sort of an electrical signal that can then be measured. For why is gamma more penetrating? Because it doesn't interact as well, because it doesn't have a mass in the same way that the alphas and the betas do, and the alphas and betas also have a charge, and so that charge can cause them to interact more with matter, whereas gammas don't, so they can penetrate through matter a lot more easily.
Wonderful. Okay, we're coming to the end of our time. So thank you so much, Katie, for your wonderful talk and also for answering all those questions so eloquently. There was a wide range of questions which you handled very expertly, so thank you very much. So all that remains for me to do is to thank Katie again. Thank you very much. Thank you, everybody who came live, and thank you, everyone who asked a question. We had some really insightful questions, and I'm sorry we didn't get to them all. I wish you a pleasant rest of your evening and see you next week. Thank you very much.